Yeah, so in today's tutorial, we're going to introduce biochemistry. Okay, then I'll also work on the tutorial sheet for biochemistry. Then I'll share the videos on YouTube as well as on the website. I'll upload the full video solution on the website. Okay, but for now, let's understand what we are going to be doing in this biochemistry course. Okay, so now we are saying biochemistry is simply the study of the chemical composition of living matter and of the chemical changes that occur during life process, right? So in other words, we are saying biochemistry, we're going to be looking at the science that attempts to explain the life in terms of the chemistry, the biology, and the two basic activity of living organism. Okay, that's what biochemistry is all about. Okay, so notice that we are going to be looking at the origin, the formation, the function, deficiency, and some of the symptoms that you're going to have in some of the biomolecules that you're going to be studying. Okay, so look at different types of biomolecules, and we're going to be looking at the source of those biomolecules, how they are going to be formed, their function, what we can have if we have deficiency of those biomolecules, and the symptoms that we can tell that there's deficiency of a certain type of biomolecule. That's what this biochemist is going to be looking at. Okay. Now, I've been mentioning biomolecules. So some of the biomolecules that you're going to look at are carbohydrates, lipids, proteins, nucleic acid, vitamins, and minerals. Okay, so in this semester, the first semester, we are supposed to cover, we're expected to cover these biomolecules. Okay, so we need to look at carbohydrates. What's the source of carbohydrate? What's the function of this carbohydrate? If you're having deficiency of this carbohydrate, what condition are we expecting to have, right? And then the same thing will apply to lipids, isn't it? How they are formed, okay? What do we expect to have if we have any abnormalities when it comes to lipids, right? We also need to understand as well. Same with proteins, nucleic acid, vitamins, and minerals. So this is what we are going to be covering this semester. Okay. So now this altogether, I call it general biochemistry. Okay. So that's what we are going to cover this semester. Now, when we go to second semester, we are going to look at the other type of biochemistry, which is going to be the metabolism of some of these biomolecules that we've talked about. Okay. But for this semester, we're going to be looking at the function, the properties, the disorders that we're going to have if we have deficiency of these biomolecules. We need to understand how they are formed and where we get these biomolecules. Okay, so that will be for this semester. Okay, yeah. So we are saying in the second semester, we'll look at the metabolism of some of these biomolecules that we discussed. And you notice that during the metabolism of these biomolecules, you end up generating our fuel, which is ATP, isn't it? So you notice that as we are metabolizing these biomolecules that we've discussed, some of them, you notice that in the process, we are going to be getting ATP. So now, what I mean when I say metabolism, so you notice that metabolism is going to be divided into two. So we're saying metabolism, we're going to divide it into two, right? So we're going to have what we call catabolism and what we call anabolism. Okay. So anyone who know what catabolism is and what anabolism is. So this we are saying we are going to cover it in the second semester. So we're just giving the overview of the entire biochemistry course. So in this semester, we expect you to understand all those biomolecules that we've talked about. We expect you to know lipids, okay, where they come from, how they are going to be produced, what's their function. We expect you to know what condition we can have if we have deficiency of them, isn't it? So those are some of the things that we expect you to know in the first semester. Now, when you go to second semester, we're saying we're going to be talking about metabolism, right? And we have discussed that we notice that during the metabolism of those biomolecules that we've made mention of, we end up generating our energy, ATP, isn't it? So now we are saying metabolism. Let's understand what metabolism is all about. So metabolism is in two parts. There's what we call catabolism and anabolism. So what is catabolism? What do we expect to see under catabolism? And let's give an example. And what is anabolism? Okay, let's start with anabolism. What is anabolism? Anyone to give it a try? Please feel free to give a contribution. Okay. So if you are talking about anabolism, you're going to be looking at the building up of complex molecule from smaller molecules, isn't it? So for example, if you look at glycogen, so let me just give an example of glycogen. Okay, so you notice that glycogen is a highly branched glucose molecule, isn't it? So we are having glucose molecules as small units, isn't it? So let's have glucose molecules as small units, right? Then we join them together. Okay, so let's say this is a glucose molecule, this is a glucose molecule, this is a glucose molecule. We join them together like that. We form a branch. We join them together like that. We form a branch, isn't it? So a branched molecule like that is going to be glycogen, isn't it? So we know that if we're having excess of glucose in the body, just from taking a meal, you're going to notice that some of the glucose that you have is going to be stored in the liver in form of glycogen, right? So now, the building up of this glycogen is what we call anabolism, isn't it? So notice that from small glucose molecules, we join them together, we form a complex molecule. And that complex molecule is a glycogen. So you notice that that process is going to be called glycogenesis, isn't it? So Genesis from the Bible, when you hear Genesis, it means at the beginning, isn't it? So we are trying to build. So whenever we are trying to build a more complex molecule in biochemistry, the process is going to be anabolism, isn't it? And a good example for anabolism, anabolic process is glycogenesis. There are a lot of them, okay? Even if you look at amino acids, okay? So amino acids, we are able to link them together, okay? So we get one amino acid, we link it together, we form a complex molecule, which we are going to call a polypeptide, right? So a polypeptide, also known as a protein, isn't it? So that is also anabolism. So we notice that we get small units of amino acids, we put them together, we form a more complex molecule, which we are going to call a protein. Okay, so that is a good example of anabolism. So we are simply converting small molecules, okay, we get small units of molecules, we link them together to form a more complex molecule, isn't it? That is anabolism. I hope people are clear on anabolism, something that you need to understand. I hope people are clear there, okay? So now we also have what we call catabolism, right? So for catabolism, you notice that it's the opposite of anabolism. So what we are going to do, we are going to be breaking down complex molecule into smaller units, isn't it? Okay, so for example, let me get a glucose molecule. So if you look at your glucose molecule, say 6-carbon molecule, isn't it? It's a 6-carbon molecule. 1, 2, 3, 4, 5, 6, right? So we're having 6 carbons there. So what we can do, we can break this glucose molecule into two molecules, right? So we can cut them, it's there, into 3 equal parts, right? So we get a 3-carbon molecule. So now a 3-carbon molecule is called pyruvate, isn't it? So you notice that if you get the glucose molecule, we undergo a process called glycolysis. Okay, so lysis is the breaking, isn't it? So whenever we use lysis in biochemistry, we're going to be using these terms. When you hear lysis, just know we're talking about breaking, right? So if we break a glucose molecule into a smaller unit, isn't it? Pyruvate, which is a 3-carbon molecule. So we are coming from a 6-carbon molecule to a 3-carbon molecule. So we undergo glycolysis, right? Then we form a pyruvate. So that is catabolism. So we are having a more complex molecule, which is a glucose molecule, right? We break it into 2 equal parts. We form pyruvate molecule, isn't it? So that process called glycolysis, right? So we are simply breaking a more complex molecule into a smaller complex molecule, isn't it? So even from the previous example that we gave on glycogen, so you notice that this glycogen, we can actually break it down back into a glucose molecule, isn't it? By cutting the branches that we formed and these bonds that we formed, right? So we are simply breaking a more complex molecule, which is glycogen. So this glycogen can undergo what we call, okay, so we are saying examples of catabolism. We have one, glycolysis, which is the breaking down of a glucose molecule into pyruvate, isn't it? To get 2 pyruvate molecules, right? We can also have, so the one I'm talking about here, where we are breaking down a more complex molecule, glycogen, into a small unit, glucose, okay? So that process is going to be called glycogenolysis, okay? Glycogenolysis. That's another good example for catabolism. So in simple terms, just to summarize, when we talk about catabolism, we're talking about the breaking down of complex molecules, isn't it? So here we are breaking, right? Catabolism, we are breaking. Anabolism, we are building. That's what we have. So this is going to be discussed more in the second semester. But for now, we need to understand what these terms, and you should be able to give examples of such anabolic reactions and catabolic reactions. I hope people are clear there. Any question? Okay. So if there's no question, please feel free to ask when you're not clear. This is the only time you can understand. Yeah, so feel free to ask. Whenever you're not clear, feel free to ask. You can even leave a message in the chat if you can't talk. Okay, yeah, so that's what we have. Now, we've said for this semester, we are focusing more on the biomolecules, and we've made mention of some of the biomolecules that we have. We have carbohydrate, lipids, protein, nucleic acid, vitamins, minerals. And remember what we're going to be looking at mostly. We want to know the source. We want to know the function, right? We want to know how we are going to be creating these biomolecules, right? We want to understand the structures of these biomolecules, right? We want to understand the deficiency of these biomolecules. What condition do we get if we are not having normal amounts of these biomolecules, right? So now let's look at some of the functions that we can have. Okay, so for example, carbohydrate, you're going to notice that they're going to be the primary source of energy. So most of the time during metabolism, we get your glucose molecule, we break it down into pyruvate, right? Okay, so you notice that in the process, as we are breaking down this glucose molecule by glycolysis, okay, notice that in the process you'll be producing ATP, right? So we are going to understand this more in semester 2. But for now, just know that carbohydrates, they're able to give us energy. Okay, so through glycolysis, even gluconeogenesis. Now, gluconeogenesis is the building up, right? Where we are coming from the pyruvate molecules, we get back our glucose molecule. That would be called the gluconeogenesis. So in gluconeogenesis, you notice that also ATP are going to be produced. So In short, carbohydrates are able to give us energy during their metabolism, isn't it? So same with lipids, right? So lipids are also a source of energy, and they also act as a storage. They are able to store energy, isn't it? So they are a large storage uh, biomolecules when it comes to energy, right? Even better than carbohydrate because of them having a lot of hydrogen atoms compared to the hydrogen atom ratio that we have in carbohydrates. Okay. Apart from that, you notice that a certain type of complex lipids are going to be part of your uh, plasma membrane, and those are going to be the phospholipids. Cholesterol is also there to help when it comes to rigidity of the cell membrane. So that's what we have there. Okay. All right. Yeah. So that's one of the function of these uh, uh, lipids, right? So we're saying they are part of the cell membrane, and some are going to be part of they are also acting as energy source, isn't it? So that's what we have there. Okay. Then we also have proteins. Their function, they are going to be most the structure and functional unit of the body, isn't it? Then we have nucleic acid, which are going to be responsible for the storage of genetic information, isn't it? So that is DNA, and also the transfer of our genetic information through RNA, isn't it? That's a function of nucleic acid there. Okay, vitamins are also essential for our our body, for our accessory growth factors. Okay, so we're also going to look at vitamins and minerals at some point. So those are some of the functions of these uh, uh, biomolecules that we are going to look at. Okay, so now if we start doing carbohydrates, you notice that you're going to give more function, and you need to know a lot of function for just carbohydrates alone. They just don't act as energy source. There are also other functions, and you need to know, right? Okay. Yeah. So we also have enzymes. Now, enzymes are most of them are proteins in nature, isn't it? So they are the ones that are going to act as catalysts, isn't it? They are going to speed up a chemical reaction by reducing what we call activation energy, isn't it? So that's what we have, right? Yesterday we were talking about exothermic endothermic reaction. We said there's that amount of energy that we need to overcome for us to get the activation energy, right? Okay, so now you notice that what an enzyme is going to do is going to lower that amount of energy that you need to overcome for you to get the product. Okay, so you notice that for a reaction with an enzyme, the activation energy will be lower. So you end up having something like that. Okay, so the amount that you need for you to start up a chemical reaction, the amount of energy will be lower compared to when you're not having an enzyme. That's what we have. Okay, so also have hormones which are going to be acting as chemical messengers. Okay, so that's what we have there. Okay, so some of the conditions that we can have if we're having deficiency of some of the biomolecules that we discussed. For example, nucleic acids. If you are not having normal nucleic acids, we end up having genetic disorders. Protein, like for example, hemoglobin is a protein, isn't it? So if you have a mutation in the subunit of your hemoglobin, right, you end up having what we call sickle cell anemia. We'll understand when we discuss them in food, isn't it? Also have lipids. Accumulation of lipids in a blood vessel is called atherosclerosis, isn't it? So a condition that can result from lipids. Carbohydrates can end up having diabetes mellitus. Okay, that's what we have there. So those are some of the conditions that we can have if you are not having normal biomolecules in the body. Right. Okay. So importance of biochemistry laboratory investigation. Now, the investigation that we are going to be carrying out, if at all you are going to do any lab this semester, notice that you're going to have two types of investigation. We're going to have what we call quantitative analysis and qualitative analysis. You need to understand what qualitative analysis are going to be when you enter in the lab and what quantitative analysis are going to be all about when you enter in the lab, right? So for qualitative analysis, you notice that that's the analysis you're going to be doing that won't be involving numericals, isn't it? So for example, we are testing for proteins, right? We say uh, uh, we use, I don't know, is it the Benedict solution, right? So we see the color change, right? So we are able to see the color change. If it's a violet, purple, violet, whatever, you are able to say, okay, there is presence of proteins or not, right? So depending on the color that you are able to see, you are not involving any numbers there. You're just observing what color you are getting, right? So that is going to be called a qualitative analysis. So no numbers involved, only you are observing using your senses, your spatial senses, right? You're just observing, okay, there's a color change here, right? Or you say, you can say there's a pungent smell. So you just having the pungent smell and you say there's presence of ammonia. That's it, a qualitative analysis, right? You don't involve any number, right? But there are going to be those analysis which will involve digits, right? So we measure a specific volume of or let's say we measure a specific weight of your sample, we get it, let's say 0.2 kg. Okay, so we are involving numbers. Okay, so those analyses that are going to involve numericals are going to be called quantitative analysis. So this one will be involving numbers, right? Okay, so we need to measure, let's say we measure 2 mils. So that's a qualitative analysis, a quantitative analysis. It involves a number, right? But for colors, smell, those ones are going to be qualitative analysis. So do not involve numericals, isn't it? That's what we have. So those are the two types of investigations that we can have in the laboratory. Okay. Any question up to here so that we revise some of the things that we discuss? Let me get questions. So we are just introducing a course. So when you this coming week, we are going to do a topic which will be water and pH, right? So that's where we start from. Then we'll go to the biomolecules now. I don't know which one. We'll start with maybe carbohydrates. We'll be moving like that. We'll go to lipids, proteins, like that. So we are just creating a roadmap of how we are going to be moving so that if we need to study in advance, should be able to study like that. So you know our targets. We need to understand carbohydrates. We need to understand lipids. We need to understand proteins. We need to understand vitamins if you are going to have enough time. Most of the time, for vitamins, you can end up doing them in semester too, depending on how much time you're going to have. But we expect you to know it is missed. Okay. Also, uric acid. Okay. So any questions up to here? Anything that you'd like? Everything is clear. Let me get a confirmation. Are we all clear here? Okay. So I don't know why people are not participating, but here for these questions, I want to know if people are following. So I've put some few questions here so that you should be able to answer. I want to know if you are following. Okay. So I hope people will participate. Okay. So feel free to answer. Let's start with the first question here. Which statement best define medical biochemistry? So the first one saying the study of chemical reaction in non-living system. The second one saying study of metabolic function in plants. The third one saying study of biochemical constituents and their interactions in the human body to maintain healthy energy production. Then study of enzyme kinetics only. So which one is the best definition?
C, sir.
Anyone?
C.
C, good. Okay, C, right? So if you look at A, the non-living organism, non-living system part is making it wrong, isn't it? The second one, we're saying study of metabolic function in plants. So this one has only covered for the second semester part, which we are saying we're going to discuss the metabolism of most of the biomolecules that we've talked about. And it's not just in plants, right? We are going to be looking at humans. We are doing for a medical course for humans, right? So we have to focus on humans. So that is also wrong, right? The study of enzyme kinetics on that's also wrong. Okay, so let's go to part 2. This is saying which of the following best describe the scope of functional biochemistry. So A saying structure analysis of biomolecules, study of metabolic pathways, understanding the function of biomolecule, underlying life processes, okay, determination of molecular weight of macromolecules. Which one is the answer there? Anyone to give it a try?
Understanding the functions of biomolecule, underlying life processes.
Good. Okay, so that's what we have for option number 2, isn't it? Okay. Then let's go to part 3. Which biomolecule acts as the secondary source and form a major component of plasma membrane? Which one is that? So we have carbohydrate, proteins, lipids, and nucleic acids. Which one is which? Okay, so now if you look at the plasma membrane, say lipid bilayer, isn't it? So it will have a phospholipid, right? One phospholipid lining the inside of this cell membrane, and the other one on the outer. So notice that, let's say we're having water content on the outer layer. So notice that the polar heads are going to be the water content on the outer layer. And then also in the inside, we're having water content here. So even here, the heads are supposed to follow where the water content is, right? So the polar one, and we're saying it's the hydrophilic one, hydrophilic water loving. That's why we're having such an arrangement. We end up having the heads facing outside, tails inside, right? Now that's a phospholipid, and that's the largest component of the plasma membrane, isn't it? Yeah. So also we are going to have cholesterol here. So the cholesterol is there to control the rigidity of fluidity of the plasma membrane, isn't it? So depending on the condition that we have, if we're having high temperature, this cholesterol, there is a way we're going to arrange it. If we're having bigger temperature, this cholesterol, there's a way we're supposed to arrange it. Okay, so now I'm going to find time before we even do water and pH. I'm going to come and discuss the plasma membrane itself. We need to understand it in detail. We need to understand all the components. And so before we even go to water and pH, I think I'll tackle that, then we can move to water and pH, right? Okay, so that's what we have there. Then we can also have proteins. Some proteins are going to be embedded, okay? Some proteins are going to be penetrating the plasma membrane. So those ones that are going to be embedded just on the surface of the plasma membrane, we're going to call them the peripheral proteins, right? And those ones that are going to be penetrating through the plasma membrane, they are going to be called integral proteins, right? So we're going to come back and discuss all that. But the largest component is a phospholipid, isn't it? Which is a lipid, right? Okay. So understand how this phospholipid is going to be formed and how it looks like and its functions apart from being the component of the plasma membrane. Okay. All right. So let's go to the last part. Enzymes are termed a biological catalyst because they are consumed in the chemical reaction. They catalyze, they alter the equilibrium of the reaction, they accelerate specific biomolecular reaction without being consumed. They provide energy to drive endothermic reaction. So what's the function of these proteins or the enzymes? So what's the answer there?
Just strength. I think the enzymes are termed as biological catalysts because they accelerate specific biochemical reactions without being consumed.
Good. So that's the answer, isn't it? So we discussed we just talked a bit about enzymes. We are going to discuss in detail. We'll look at their inhibitions, most the inhibitions, because those are the ones that usually take us to drugs, right? As pharmacists, right? But we'll discuss those things at a later stage. But for now, we know that the function of this enzyme is to speed up a chemical reaction by lowering activation energy, isn't it? And in the process itself, it's not going to be consumed. So it's there to act as a catalyst, right? So that's what we have. Okay, thank you so much. Very good. Okay, so that's what we have for today. We've just introduced biochemistry. We are going to continue in the next tutorial. The next time we meet, we'll discuss the plasma membrane. Now I'll have that class before Wednesday. Wednesday, that's when we are supposed to have our biochemistry. We didn't have this one, so this was a maker. So on Wednesday, before Wednesday, I'll put another class so that we just discuss the plasma membrane, we understand it, then we can go to water and pH. And I'll also start working on the tutorial sheet. But for the tutorial sheet, you are going to access it on the website. So I'll show you how you can be accessing the tutorial questions once I upload them on the website. Just on this topic, there are tutorial questions that I'm going to upload there. So if you are registered, you should be able to access them on the website. I'll send the video when I am done working on those tutorial questions. Okay, so if there are no other questions, we can end here. Thank you so much.